Q.1 Q5 Multiple choice question, MCQ, carry 1 mark each, for each wrong answer, 1/3. Q.1 Antony van Leeuwenhoek observed several microscopic organisms under his handmade microscope. He described them as A. Bacteria, B. Fungi, C. Animalcules, D. Bacteriophages. Q.2 Which one of the following pathways oxidizes 1 mole of glucose to 2 moles of pyruvic acid along with 1 mole each of ATP, NADH and NADPH, in Pseudomonas spp, but not in Bacillus spp? A. Gluconeogenesis. B. Embden-Meyerhof pathway EMP. C. Endner-Duderoff ED pathway. D. Pentose phosphate pathway PP. pp. Q.3 Water balance in extreme halophiles such as Halobacterium is maintained by cell surface glycoproteins consisting of A. Glycine and lysine, B. Lysine and histidine, C. Glycine, D. Aspartate and glutamate. Q.4 Nocardia spp. Are not amenable to the classical method of Gram staining due to the presence of A. N-acetyltyrosineuronic acid in the cell wall, B. Thick peptidoglycan, C. Mycolic acid, D. Ketodeoxyoxylisonic acid. Q.5 Protists belonging to the genus Trichonympha thrive in the gut of termites. They help the termites use wood as a food source. This relationship is an example of A. Parasitism, B. Competition, C. Commensalism, D. Mutualism. Q.6 Q9 Multiple select question, MSQ, carry 1 mark each, no negative marks. Q.6 Which of the following is used as electron donor S for CO2 reduction during photosynthesis in purple sulfur bacteria? A. Hydrogen sulfide, B. Thiosulfates, C. Methane, D. Sulfates. Q.7 Which of the following catalyzes substrate-level phosphorylation? A. ATP synthase, B. Succinate thiokinase, C. Phosphofructokinase, D. Pyruvate kinase. B. Succinate thiokinase, C. Phosphofructokinase, D. Pyruvate kinase. Q.8 Which of the following methods can be applied to identify a bacterial species? A. Fluorescent in situ hybridization FISH. B. Polymerase chain reaction PCR, followed by sequencing of the amplicon. C. Gram staining. D. Acid-fast staining. Staining. Q.9 Which of the following events would contribute to the induction of lac operon in a wild-type strain of E. coli? A. Accumulation of allolactose in the cell. B. Direct binding of CAMP to the promoter DNA. C. Binding of CAMP to a specific protein leading to its interaction with the promoter. D. Elimination of CAMP from the cell. Q.10 Numeric Numerical answer type NAT carry 1 mark each, no negative marks. Q.10. 1 mole of a circular bacterial plasmid was digested with a high-fidelity restriction enzyme. The plasmid has 5 restriction sites for the enzyme used. The number of moles of fragments released upon cleavage at all sites is _blank. Q.11 Q16 multiple choice question, MCQ carry 2 mark each, for each wrong answer, 2/3. Q. 1. Under anaerobic fermentative growth conditions, 1 mole of glucose yields 22 grams of Streptococcus faecalis or 8.6 grams of Zymomonas mobilis. The molar growth yield YATP for I S faecalis and E Z mobilis will be A. I. 11 and E. 4.3, B. I. 22 and E. 4.3, C. I. 22 and E. 8.6, D. I. 11 and E. 8.6. Q. 12. The order of abundance of quinones ubiquinone UQ, menaquinone MQ, and dimethylmenaquinone DMQ in E. coli growing anaerobically on fumarate is A. UQ greater than DMQ greater than MQ. B. MQ greater than DMQ greater than UQ. C. MQ equals DMQ greater than UQ. D. MQ greater than UQ greater than DMQ. DMQ. Q. 13. What is the number of ATPs generated per molecule of NADH during oxidative phosphorylation in E. coli via I NDH1 and cytochrome BO complex or E BNDH2 and cytochrome BD complex? Assume H plus ATP equals 3. A. I 2.00 and E 3.67. B. I 3.00 and E 2.67. C. I 2.70 and E 0.67. D. I 2.50 and E 0.50. .50. Q. 14. Match the immunoglobulin classes with their function. Immunoglobulin. Function. I. IgA. P. Protects the fetus. E. Ig. Q. First antibody to be produced in response to infection. E. IgM. R. Provides localized protection of mucosal surfaces. IV. IgA. S. Mediates allergic reaction. T. Directly lyses the target cells. A. I. S. E. P. E. Q. IV. R. B. I. P. E. T. E. Q. IV. R. C. I. Q. E. P. E. T. IV. R. D. I. R. E. P. E. T. IV. S. Q. 15. The figure shows the profiles of quantitative real-time PCR (qRT-PCR) tests for SARS-CoV-2 conducted on the throat swab samples of 3 individuals X, Y and Z. Tests were carried out under identical conditions. Dotted line represents the threshold fluorescent value. Identify the correct statement on the status of the COVID-19 tests of the individuals based on their qRT-PCR profiles. A. X and Y are negative, Z is positive. B. X and Y are positive, there is no apparent difference in the viral load. C. X and Y are positive, X has the highest viral load. D. X and Y are positive, Y has the highest viral load. Q.16. The rate of appearance of recombinant E. coli strains containing different genes after a mating between HFR and F strains is shown in the graph left. The approximate location of different genes page, Q, R, S, T, X, and Y along the HFR chromosome is also shown right. Based on this information, identify the recombinants X, Y and Z. A. X is X plus, Y is R plus and Z is P plus. B. X is P plus, Y is R plus and Z is X plus. C. X is X plus, Y is P plus and Z is R plus. D. X is P plus, Y is X plus and Z is R plus. Plus. Q.17 Q20 Numerical answer type NAT carry 2 mark each, no negative marks. Q.17 The genome of a bacterium encodes for 10 different surface antigens, whose expression can be turned on or off randomly and independently. The number of possible antigenic combinations is _blank. Q.18 Suppose the mRNAs in a newly discovered bacteria are composed of only 2 distinct nucleotides, as opposed to 4 found in all known organisms. Considering that the organism has no nucleotide modification systems, the number of nucleotides required per codon to encode at least 20 distinct amino acids will be _blank. Q. 19. The decimal reduction time d